Hello, welcome back to the sixth episode of this series. Finals are coming up for me, so please bear with me as I deal with this schedule. But today we're going to graduate from single neurons into the connected systems that make up our bodies. Neurons need ways to encode information for communication, and neuroscientists need ways to quantify this encoding, so we'll start to cover both in this video. By the end, you should be able to answer the questions: how do neurons apply grid and action potentials for encoding purposes, as well as how can scientists graph neuron activity? Like I said in the intro, we're going to start to look at systems neuroscience, which follows the structure and function of interconnected neurons. Interconnected neurons, in turn, make up neuronal pathways, circuits, and networks, three terms that I may use somewhat interchangeably. Neurons work in networks to transmit, process, and respond to information. Often, these networks are named or described by the type of information they're generating. A lot of the time, these systems also heavily use a single type of neurotransmitter as well, so that may also be a way of people classifying it. Networks will act on various scales as well. Micronetworks connect small populations of neurons and often have simple patterns or outputs. These can then join up into macronetworks, doing things like connecting disparate brain regions together. Complex thinking and motor skills are just some examples of the behaviors that will be really hard to do without the use of macronetworks. Information encoding can be done by a number of tactics, and R2 potentials can play a part. In, say, a sensory neuron, we need to be able to take a stimulus and convey that with meaning with some level of amplitude and duration as well. Graded potentials can help transduce a stimulus to an electrical response, acting as a rough correlate of amplitude and duration. Enough of these will add up, and you can get an action potential shooting down the chain. But it's not like a stimulus ends with just one action potential or spike. Many action potentials can be fired. It would be really hard to tell between different types of stimuli if every time a hair on your arm was triggered, the same pattern was sent. No, just like considering combinations of cells, individual neurons can send combinations of action potentials, kind of like bits in a computer, to feed info about duration of the stimulus, the length it was applied, and maybe some more features of if the neuron is specialized for that, like, say, heat. Let's get started with the first quiz question. Reflex arcs often only consist of a sensory neuron, interneuron, and motor neuron, along with their associated receptor and effectors. Which of the following best describes a reflex arc? A. Synapse, B. Dendrite, C. Macro network, or D. Micro network? Remember, we're up in systems world now, so answers pertaining to single neurons are not going to be the answer. A and B are out for this reason. The question only calls 3 neurons, which is quite small scale. Therefore, the circuit is best described by being a micro network. Therefore, D is the best choice. Neuroscientists have a remarkable number of tools to visualize the electrophysiological changes happening during information encoding. So let's turn down a couple of ways that we can look at and graph these data points. Raw membrane voltage recordings can be plotted against time to show a simple, unrefined graph of every time an action potential goes off. You can directly see each action potential as a spike on the recording. A whole combination of these spikes is called a spike train. You do have to be careful with this raw data because it's possible for noise to sneak in, so cleaning your data is crucial. A slightly more sophisticated graph is a raster plot. This also shows every spike, but they're represented simply as a dot. A y-axis can expand to accommodate different trials, allowing for easy comparison between recordings. Oftentimes, you'll see the application of the stimulus being demarcated so you can visualize the change from baseline as well. If we're not particularly interested in individual spikes, but rather greater patterns of firing, histograms do well by placing a bunch of spikes into a bucket for a given time interval. Peristimulus time histograms, PSTH, are similar to what we were discussing, but once again are provided with buckets of time intervals. Alternatively, interspike interval plots uniquely plot the time between action potentials instead of the potentials themselves. All the different graphs we talked about fall out of the raw data, so it's important to keep the measuring instruments as noise-free as possible. Back in Neurolab, we had some gnarly ways to do that. Like, we would be running experiments on cockroaches, but their being alive would kind of pose a confound. So we would either remove their heads or their legs just so we can do our tests without any outside information coming in. I'll put up some clips if I can find them, but you might want to look away for a bit if you don't like these creepy crawly. All right, the bugs are no longer on the screen, so let's take some time to do another practice question. If you administered a drug that decreased the amount of action potential sent over a given time, which of the following changes would you expect to see on the graphs? A. More dots on the raster plot after administration. B. higher bars on a peristimulus time histogram, assuming the same axes. C. Longer interspike intervals on an interspike interval plot. Or D. Shorter slanted spikes on the spike train. This is a tricky one, but the answer is C. Answer choice A is wrong because more dots on the raster plot means more action potentials, which is the opposite of the drug's effect. Similarly, with answer choice B, higher bars in the PSTH also correspond to more action potentials being sent. Answer choice D is tricky because we have to go back to think about what a spike train even shows. It shows action potentials, which, while being less in number, don't lose out in their speed or change of their voltage. They're very all or nothing. So there's just less spikes in the train, but the individual spikes themselves are largely unchanged. Answer choice C is correct because there's less action potentials, meaning there's more time in between them, meaning the ISI plot would reflect the change as well. All right, that brings episode 6 to a close. We started to talk about the basics of information encoding and how you can measure it in the lab. When we tackle the senses in a later unit, we'll be able to bring this knowledge back to specific applications. As always, timestamps in the description. Thank you for watching, and I hope to see you next time.